welcome to MYP Biology. Today our topic is going to be about enzymes. So let's find out what are the enzymes. So what is the link between digestion, photosynthesis, bread wine, and washing powder? What do you think? They all depend on enzymes. So enzymes are biological catalysts. They speed up the chemical reactions that take place inside our cells, but without being used up in the process. There are many thousands of different types of enzymes, and each of one catalyzes a different reaction. Enzymes occur naturally in all organisms, but they are increasingly being used in industrial processes. As you see here, enzymes are protein molecules and so are made up of amino acids. Most enzymes contain between 100 and 1,000 amino acids. These amino acids are joined together in a long chain which is folded to produce a unique 3D structure. The shape of an enzyme is very important because it has a direct effect on how it catalyzes a reaction. Why do enzymes have a different shape? An enzyme's shape is determined by the sequence of an amino acid in its structure and the bond which forms between the atoms of those molecules. Different types of enzymes have different shapes and functions because the order and type of amino acids in their structure is very different. So you're going to answer this about the enzymes true or false. How do enzymes work? What are enzymes so specific in their reactions? Enzymes are very specific about which reactions they catalyze. Only molecule with exactly the right shape will bind to the enzyme and react. These are the reactant or substrate or a molecule. So each enzyme has actually the site which is called the active site which the reactant binds. So here there's a very specific shape and the most important part of the enzymes. Look at this picture. It's the same way that a K fits into a log. So a substrate is thought to fit into an enzyme's active site. So this enzyme is the log and the reactant is actually the key. So look at this. The enzyme is actually playing a role of a log, but the reactant is actually the key. So enzyme plus reactant, enzyme reacting complex, enzyme plus product. So here actually is the log and key model. Enzymes speed up reaction by lowering the activation energy of a reaction. So enzyme reactions really depend on a lot of things. So several factors include temperature, pH, enzyme concentration, substrate concentration, surface area, and the pressure. All enzymes work best at only one particular temperature and pH. This is called the optimum. So if the temperature and pH change sufficiently beyond an enzyme's optimum, the shape of the enzyme irreversibly changes and it becomes denatured because they are actually the proteins. This affects the shape of the active site and means that the enzyme will no longer work. So their optimum pH and temperature is very important. So if it's very cold or if it's very hot, the enzyme cannot work properly. So at the end of this process, they are actually denatured. So this is the here. Here we're going to identify the enzyme terms. Here, enzymes in life. Enzymes catalyze the thousands of reactions that need to take place in order to maintain life. So what are some of these reactions? Digestion, respiration, photosynthesis, and protein synthesis. Not all enzymes work inside cells. In what processes do enzymes work outside? Digestive enzymes are produced by specialized cells in the pancreas and digestive tract. These enzymes pass out of the cell into the stomach and the small intestines, and here they help the digestive system. For example, as food moves through the digestive system, it is broken down and useful food molecules and water are absorbed into the body. After food is swallowed, it enters the stomach. Then two things happen to the food in the stomach. The chemical breakdown of it begins. microbes are destroyed. From the stomach, food enters the small intestine where digestion is completed and the small digested food molecules are absorbed into the bloodstream. Here, digestive enzymes found in the small intestine are damaged by a strongly acidic pH. So that's why the liver produces bile, which is an alkali, which is stored in the gallbladder and released into the small intestine. So as you see here, the bile neutralizes the acidic contents coming from the stomach, creating the alkaline environment that the intestinal enzyme need to work. So here we've got the body carries out digestion, so we have got carbohydrate, protein, and fat molecules to be broken down. So those are the large food molecules. So how are they going to be broken down? Starchy food contains carbohydrates, which are made of long chains, and the body breaks down long chains of carbohydrates into a smaller sugar. They get the energy. What about the protein molecule as well? And it comes the fat molecules as well. So they are made of glycerol and fatty acids. So the carbohydrate, protein, and the fat molecules need to be broken down to be absorbed by the body. So here we have different type of the enzymes here to be break down all large molecules. So carbohydrates breaks carbohydrate into smaller sugars. Protease breaks protein into amino acid, and lipase breaks fat into fatty acid and glycerol. So look at this one, how carbohydrates breaks the carbohydrate chain. So non-carbohydrate molecule are undergoing the carbohydrates enzyme, so they actually produce the sugar molecules at the end of the just breaking. The protease break down the long protein molecule into small amino acid molecules. The fat digestion, the bile and the lipase actually help it. The bile actually help the emulsification, but the lipase actually take it into the glycerol and fatty acids. They are the example of the enzymes, the substrate, enzyme, and the products that you have to learn this one. This is actually the game. You're gonna just match the enzymes with their products. Here we've got a video. Let's watch a video about digestive enzymes.
We don't have chunks of cake or bits of burger floating around our bloodstream. So how do we get the energy from our food to our cells? Two things have to happen before we can make use of our lunch. It needs to be broken down into smaller pieces or digested, and then it needs to be absorbed into the bloodstream. Digestion takes place in lots of places along the alimentary canal, and absorption happens in the small intestine, which is found just after the stomach along the length of the digestive system. To learn more about the digestive system, watch this video, and to learn more about absorption, watch this video. In this video, we're going to look at the enzymes involved in digestion in more detail. The large molecules which make up our food, like lipids, proteins, and carbohydrates, are too big to be moved into our blood. So, they need to be digested into smaller molecules by physical processes like chewing, and chemically by special proteins called enzymes. Different types of enzymes digest the different types of food, so let's look at the food groups in turn. First, carbohydrates. Foods like rice and pasta are made of carbohydrates. The simplest carbohydrates are sugars, which can be joined into big chains to make complex carbohydrates, like starch. Carbohydrates are digested by carbohydrase enzymes, such as amylase. Amylase is a special type of carbohydrate, which breaks down starch, a big carbohydrate, into smaller molecules. These can then be broken down further into glucose, which is small enough to be moved into the blood. Amylase is found in your saliva, which is where carbohydrates will first start to be broken down chemically, and another amylase is released into the small intestine from the pancreas, so much further down your digestive system. Now let's look at proteins. Proteins are found in meat, fish, beans, and pulses. They are made up of amino acids and are digested by protease enzymes. The protease enzymes break down proteins into amino acids. Protease, called pepsin, breaks down proteins in the stomach, but this doesn't work once the food moves into the small intestine. In the small intestine, the conditions are different. So a different protease called trypsin is released into the small intestine from the pancreas for continued protein digestion. What conditions do you think might be different in the small intestine compared to the stomach? Whilst the stomach is very acidic and has a pH of about 2, the small intestine has a higher pH of about 8. Protease enzymes, which work well at pH 2, don't work at pH 8, which is why different protease enzymes are needed in the small intestine. And the last group of food, lipids. Lipids are digested by enzymes called lipases into glycerol and fatty acids, but it's not an easy job. It requires a process called emulsification to take place first. Emulsification breaks the lipids into smaller droplets. The smaller droplets have a larger surface area for the lipase enzymes to work on. To learn more about bile and emulsification, watch this video. So, to recap, the main enzymes involved in digestion a carbohydrate called amylase is released in the saliva. This digests starch into smaller sugars. A protease called pepsin digests protein in the stomach. Then a protease called trypsin further digests proteins into amino acids in the small intestine. A carbohydrate called amylase and lipase are also released into the small intestine from the pancreas. They digest carbohydrates into sugars and lipids into fatty acids and glycerol. These molecules are small enough to be absorbed into the blood.
So now let's see how enzymes work in industry. How many items are made using enzymes? So bread and wine and alcohol are made by the use of the enzymes. So a confectionery, a type of enzyme called isomerase converts the sugar glucose into fructose, another type of sugar. Fructose is sweeter than the glucose, so a smaller amount is needed. This makes fructose syrup a useful ingredient in slimming foods. Invertase is used to create soft center chocolate. The center initially contains sucrose and is hard. So chocolate, of course, is also made by the use of the enzymes. How do enzymes help to clean clothes? Of course, we have some kind of dirty clothes that they have been stained by the very greasy, so some food are actually got into that. So the enzymes are coated with a special wax that melts in the wash, releasing the enzyme, and those enzymes actually get on the clothes surface and they actually clean all the clothes by the washing powder. So the protease, lipase, and carbohydrates actually play an important role in actually washing and cleaning the clothes as well as here. Many of the enzymes used in the home and in the industry are naturally produced by microorganisms such as bacteria and yeast. So bacteria and yeast can be manipulated or genetically engineered to produce these enzymes, and this called this fermentation. So how are the enzymes produced? Here we've got the glossary, the words that and definitions that you don't know about this. There's the match the reactant and the homework worksheets about digestive enzymes and substance involved in digestion, which you're going to do at home. Thanks for watching, guys. Have a nice day. Bye bye.